Hello friends around the world, and welcome to the Caravan to Midnight pre-show. I am Dr. Lawrence Sellin, sitting in for John B. Wells. Since the onset of the COVID-19 pandemic, I've been investigating three areas: the origin of COVID-19, the structure of China's biowarfare program, and the 40-year massive infiltration of U.S. research laboratories by Chinese Communist Party scientists, including members of the People's Liberation Army, which directly contributed to China's biowarfare program and the laboratory creation of COVID-19. Despite the continuous Chinese Communist Party propaganda claiming that COVID-19 was a natural transmission from animals to humans, the mindless regurgitation of that view by the American mainstream media and the U.S. government's premeditated uncertainty about the origin of COVID-19, the preponderance of scientific evidence indicates that it was man-made in China. First, it is important to note that after 2 years of extensive research, those claiming that COVID-19 had a natural origin have not provided any significant evidence to support that contention. In contrast, COVID-19 possesses a number of unique features which makes a natural origin highly unlikely. Foremost among those is a small structure within the virus called the polybasic cleavage site, which can be considered the smoking gun evidence of the laboratory origin of COVID-19. The polybasic cleavage site in COVID-19 is a 4-amino acid sequence, PRRA, a structure that is not found in any animal coronavirus from which COVID-19 could have evolved, or the human coronavirus that caused the 2002 pandemic. Furthermore, the RR part of the PRRA polybasic cleavage site is coded by the gene sequence CGG, an extremely rare double codon also not found in any coronavirus from which COVID-19 could have evolved. It has been known for over 20 years that the presence of a polybasic cleavage site in coronaviruses increases their transmissibility and lethality. The polybasic cleavage site also enhances COVID-19's ability to infect multiple human organ systems, including the lungs, heart, kidneys, brain, and the gastrointestinal tract. Chinese scientists at the Wuhan Institute of Virology and scientists of the Chinese People's Liberation Army have published scientific articles inserting and genetically manipulating polybasic cleavage sites in viruses. These facts alone prove that the COVID-19 polybasic cleavage site is not a product of natural evolution and was artificially inserted into a coronavirus by Chinese scientists to increase its transmissibility and lethality. In addition, during the course of the pandemic, mutations have occurred in the COVID-19 polybasic cleavage site, making it more polybasic, which likely contributed to the increased transmissibility seen in the Delta and Omicron variants. There can be little doubt that COVID-19 was part of China's advanced and extensive biowarfare program. COVID-19 aligns well with China's military doctrine. It is a highly contagious, low lethality, designed biotechnology weapon capable of being deployed in the absence of war to obtain strategic advantage for China without the destructive effects of conventional or nuclear weapons. According to Chinese military doctrine, a primary consideration in the choice of a bioweapon is plausible deniability. In the People's Liberation Army's own words, bioweapons should be used in a way that can avoid global condemnation. Bioweapons should be deployed covertly, and if suspected, the biowarfare agent can be disguised in such a way that even the most sophisticated analysis would be inconclusive as to its origin. That is, the bioweapon can be blamed on nature, just like COVID-19. All of China's research and development resources have been marshaled for biotechnology dominance. In 2016, the Chinese Communist Party's 13th five-year plan, mandated what had been an unofficial policy, the fusion of military and civilian research. Up until then, there had been unofficial but de facto biowarfare cooperation between the military and civilian sectors. China's biowarfare program exists at three levels. There is a core secret military level, which is composed of military research institutes and military hospitals. The second level includes all Chinese universities and, quote, civilian research centers like the Wuhan Institute of Virology. That second level allows the core secret military level to access knowledge, skills, technology, and research funding from the third level of China's biowarfare program, international universities and research centers, particularly those in the United States. People's Liberation Army scientists also affiliate themselves with second-level Chinese universities, often agricultural and civilian research centers, which allow them to collaborate with and sometimes work in U.S. research laboratories, while deliberately hiding their true positions as members of China's military. The three-level structure of China's biowarfare program has created a massive support network of scientists and research centers, both inside China and connected to Chinese scientists in the United States, most of whom have remained loyal to the Chinese Communist Party and maintain active research collaboration with laboratories in China, including those of the People's Liberation Army. The origin and traditional core of the People's Liberation Army's biowarfare program is not medical, but veterinary, and to some extent agricultural. During World War II, the Imperial Japanese Army had a biological warfare research center in Manchuria called Unit 731. It was officially known as the Epidemic Prevention and Water Purification Department of the Kwangtung Army. There was a second center, Unit 100, officially known as Kwangtung Army Military Horse Epidemic Prevention Workshop. When the People's Liberation Army subjugated China in 1949, the Chinese Communist Party adopted the Japanese approach, incorporating biowarfare within veterinary research. Warhorse Health Institutes, or similar centers, were established in Beijing, Chengdu, Guangzhou, Nanjing, and Yunnan, roughly corresponding with the People's Liberation Army's regional military commands. In 1953, the 5 People's Liberation Army veterinary universities were merged into a single large institution in Changchun, a mere 140 miles south of the former location of Unit 731. While Wuhan, and in particular the Wuhan Institute of Virology, have received the most attention as the epicenter of the pandemic, the research leading to COVID-19 was done in a number of military-controlled facilities in a combined pathogen development vaccine production program. There was a large virus collection effort being conducted both in China and internationally and led by the Military Veterinary Research Institute in Changchun. It is that virus collection program which provided the coronavirus backbones for COVID-19 production, as well as intermediate artificial virus constructs like RATG13, wrongly but widely cited as a naturally occurring ancestor of COVID-19. The most interesting coronaviruses were selected for further investigation and distributed to a number of different laboratories under the supervision of the Academy of Military Medical Sciences in Beijing. The Wuhan Institute of Virology, laboratories of Wuhan University, and the military hospital in Wuhan were among the centers involved in that program. The growth of China's biotechnology sector and biowarfare program was dependent on accessing knowledge, skills, and technologies from outside China. particularly from universities in the United States, which would be eager to collaborate with China and accept Chinese Communist Party money. To achieve that end, Chinese Communist Party leader Deng Xiaoping and U.S. President Jimmy Carter in 1979 signed an historic agreement for science and technology exchange, of which the China-United States Biochemistry Examination and Application Program was a part. That program involved over 60 U.S. universities, and soon afterwards, thousands of Chinese students and scholars began flooding into the United States, many obtaining permanent positions and becoming U.S. citizens, but maintaining allegiance to the Chinese Communist Party. In a process I call scientific chain migration, successive waves of Chinese Communist Party and People's Liberation Army scientists would establish themselves in U.S. research institutions. They would then invite other scientists from China into their U.S. laboratories to access American knowledge, skills, technologies, and U.S. government funding, which in turn would be fed into China's research and development programs, including those of China's military. President Bill Clinton expanded the Carter-Deng Xiaoping agreement, openly inviting Chinese military scientists into U.S. Department of Defense research centers, including the U.S. Army Medical Research Institute of Infectious Diseases at Fort Detrick. In 2011, President Barack Obama extended indefinitely the Carter-Deng Xiaoping Scientific Exchange Agreement. Today, U.S. research laboratories are de facto extensions of the Chinese Communist Party's fused military-civilian research program, in which Chinese scientists working in the United States work with scientists in China, including those connected to China's biowarfare program. In essence, China has colonized U.S. research programs, colonies that were financed by U.S. taxpayers, likely amounting to billions of dollars. Today, there is virtually nothing that happens in U.S. research laboratories about which China's military is not intimately aware. It is impossible in this short presentation to expose the thousands of Chinese scientists working in the United States who are contributing to the growth of China's biotechnology sector, but a few cases might be illustrative. Dr. Shibu Zhang, now professor and director of the Institute of Medical Microbiology at China's Fudan University in Shanghai, obtained his master's and medical degrees from the First and the Fourth Military Medical University of the People's Liberation Army. Between 1987 and 1990, he received his postdoctoral training at the Rockefeller University in New York. Since 1990, Shibu Zhang has worked in or has been associated with the Lindsley F. Kimball Research Institute of the New York Blood Center. During his over 20 years in the United States, Shibu Zhang developed an extensive network of collaborative research with other U.S. virus research laboratories and received more than $17 million in U.S. research grants, the vast majority coming from Dr. Anthony Fauci's National Institute of Allergy and Infectious Diseases. During that entire period, Shibu Zhang maintained extensive collaborative research with the People's Liberation Army laboratories, while simultaneously inviting into his U.S. laboratory and training scientists linked to the Chinese military. While attending the People's Liberation Army's first military medical university, Xu Wen Lu and Shibu Zhang together published an article on the human immunodeficiency virus HIV in 1986. After obtaining his position with the Kimball Research Institute in 1990, Shibu Zhang maintained his relationship with the first military medical university. By 2002, Xu Wenlu joined Shibu Zhang at the Kimball Research Institute while also continuing his affiliation with the first military medical university. The onset of the first SARS coronavirus pandemic in 2002 caused a major shift in the virus research activities of the People's Liberation Army and the activities of Shibu Zhang and his Chinese colleagues working in U.S. laboratories. China's People's Liberation Army became deeply involved in coronavirus research in China, but also with People's Liberation Army and other Chinese scientists who had migrated to and were working in U.S. virus research laboratories. At the same time, People's Liberation Army strategists began writing articles about new forms of biowarfare. Already in 2005, Colonel Jiwei Guo of the 3rd Military Medical University in Chongqing described a new type of bioweapon. Colonel Guo rejected the clumsy traditional biological weapons that produce mass destruction, ones that, in his words, depend on microbiology, especially bacteriology, which uses destructive bacteria, virus, and toxic living bodies obtained directly from the natural world. These weapons are subject to nature, are difficult to control, and have irreversible effects. China would use biotechnology to create new forms of designed biotechnological weapons that would be controllable and recoverable, for which China had sole possession of the vaccine or antidote. Such weapons would be highly contagious, but of low lethality and capable of being deployed under pre-war conditions. Although artificially created, the new bioweapons would retain plausible deniability, that is, could be attributed to a disease of natural origin. Non-lethal biotechnology weapons became a central theme in China's military doctrine. Yuzhen Zhao of the People's Liberation Army State Key Laboratory of Pathogen and Biosecurity in Beijing became a key link between Shibu Zhang and the People's Liberation Army's research on coronaviruses. Yuzhen Zhao was a longtime research collaborator of Shibu Zhang. Two scientists from Yuzhen Zhao's military institute would work in Shibu Zhang's U.S. laboratory. The first was Yuxian He, who initially followed Shibu Zhang to Rockefeller University and then to his laboratory at the Kimball Research Institute. The second was Lang Ying Du, allegedly Yuzhen Zhao's wife, who is still an employee at the Kimball Research Institute in New York, and in 2020 received a 5-year grant totaling $4.1 million from Dr. Anthony Fauci's institute. After the onset of the 2002 SARS pandemic, Shibu Zhang shifted his research focus from HIV to coronavirus and greatly expanded his work with the People's Liberation Army Research Institutes, including the Nanjing Military Command, which, according to Chinese whistleblower Dr. Li Mengyan, isolated the bat coronaviruses that provided the backbones for the creation of COVID-19. Shibu Zhang also began building his connections with other U.S. virus laboratories, some of which would play roles in the research leading up to the COVID-19 pandemic, namely the Laboratory of Infectious Diseases and Vaccine Research Center of Anthony Fauci's National Institute of Allergy and Infectious Diseases, Dr. Ralph Barrick's Department of Epidemiology at the University of North Carolina, a laboratory known for its gain-of-function research on coronaviruses, the Virology Division of the United States Army Medical Research Institute of Infectious Diseases, at Fort Detrick, Maryland, and the departments of microbiology and immunology at the University of Texas Medical Branch in Galveston. In 2015, Shibu Zhang was part of a joint project linking the University of North Carolina, the University of Minnesota, and the Wuhan Institute of Virology on the subject of bat-to-human coronavirus transmission. From 2012 to 2020, Shibu Zhang published 12 scientific articles with the Wuhan Institute of Virology. Between 2013 and 2020, Shibu Zhang published 11 articles with the University of Texas Medical Branch in Galveston, home of the Department of Defense-funded Center for Biodefense and Emerging Infectious Diseases, which houses a biosafety level 4 high containment facility for research with dangerous viruses, a location that has long been an infiltration target of the Chinese military. Not surprisingly, one does not need to dig very deep to find that the University of Texas Medical Branch in Galveston has been infiltrated by scientists of the People's Liberation Army, some believed to be linked to China's biowarfare program. A key connection between the University of Texas Medical Branch in Galveston and the People's Liberation Army's veterinary research centers is Pei-Yong Shi, trained in the People's Republic of China and now a professor in the Department of Biochemistry and Molecular Biology in Galveston. Pei-Yong Shi has also been an honorary professor and research advisor at the Wuhan Institute of Virology since 2007 and received more than $5 million in research funding from Anthony Fauci's institute. One of Pei-Yong Shi's links to the Chinese military's veterinary research centers in Jilin province is Hualei Wang. Both Pei-Yong Shi and Hualei Wang are listed as co-authors of a scientific article published on October 6, 2020, but Hualei Wang hides his military affiliation, which does appear in a separate 2018 Chinese publication. Hualei Wang is shown in a photo with General Xianshu Xia, one of the leaders of Chinese military's veterinary research centers. Both Xianshu Xia and Hualei Wang appear together as co-authors with Pei-Yong Shi on a COVID-19 scientific article published on January 20, 2021. They are both associated with the People's Liberation Army's Military Veterinary Research Institute. It is important to repeat that the People's Liberation Army's Military Veterinary Research Institute is believed to be an important component of China's biowarfare program. Nevertheless, in August 2020, the University of Texas Medical Branch in Galveston was designated by Anthony Fauci as one of the 10 centers for research in emerging infectious diseases and newly funded by Fauci's Institute for $82 million. Perhaps not coincidentally, Fauci also designated Peter Daszak's EcoHealth Alliance, a longtime collaborator with the Wuhan Institute of Virology, as another center for research in emerging infectious diseases, awarding the EcoHealth Alliance $7.5 million. The transfer of American knowledge, skills, and technology is not restricted to biowarfare capabilities, but also affects the commercial sector. The tale of Xian Gao is illustrative of that point. Xian Gao received his bachelor's and master's degrees from Nanjing University in 1985 and 1988 respectively. As noted earlier, Nanjing, headquarters of the People's Liberation Army's Nanjing Military Command, was involved in some of the key research leading to COVID-19. Xian Gao completed his PhD in 1994 at Thomas Jefferson University in Philadelphia. Before returning to China in 2000, he received 6 years of postdoctoral training in the United States at the Roche Institute of Molecular Biology, the Jackson Laboratory in Maine, and the University of North Carolina. The last being the institution of Ralph Baric, known for his coronavirus gain-of-function research and collaboration with the Batwoman, Zhengli Shi of the Wuhan Institute of Virology. The most consequential time Xian Gao spent in the United States was his 2 years at the Jackson Laboratory, the world source of more than 8,000 strains of genetically defined mice and home of the Mouse Genome Informatics database. Xian Gao would subsequently reproduce the Jackson Laboratory at Nanjing University as its director of the Institute of Model Animals. Genetically altered mouse models, a biotechnology pioneered by the Jackson Laboratory and now widespread in China's military research centers, was a technique critical to the creation of COVID-19. Pre-adaptation for human infection can be achieved in a laboratory by repeatedly exposing bat coronaviruses to genetically engineered mice that express the human angiotensin-converting enzyme 2 receptor, a process known as serial passage. Xian Gao was also the founder of a Chinese company, Gem Pharmtech. Gem Pharmtech was partially funded by the Chinese Communist Party, as well as receiving investment from the U.S. capital management firms Morgan Stanley and Sequoia Capital. Gem Pharmtech opened a U.S. operation where it will house more than 6,000 mouse models in Massachusetts and California, directly competing with the Jackson Laboratory and other U.S. companies. It is no coincidence that the Jackson Laboratory filed a suit in September of 2017, alleging that Nanjing University in China was stealing its technology, breeding and reselling a strain of mice that the Jackson Laboratory had developed. I began this presentation by identifying 3 areas of investigation: the origin of COVID-19, the structure of China's biowarfare program, and the massive infiltration of U.S. virus research laboratories by Chinese Communist Party and People's Liberation Army scientists. I have demonstrated that those 3 areas are not separate, but connected to each other. The development of China's biowarfare doctrine occurred in parallel with the expansion and fusion of the military and civilian research sectors, the chain migration of Chinese Communist Party and People's Liberation Army scientists to the United States, and the acquisition by them of cutting-edge biotechnology knowledge and techniques, all of which contributed to the creation of COVID-19. We must now recognize that China has misused all the agreements for science and technology exchange it has made with the United States and exploited open scientific collaboration to gain economic and military dominance over the United States. All funding involving scientists from the People's Republic of China who are presently working in the United States, and all funding related to collaborative research with scientists in China must be immediately halted until a comprehensive national security evaluation is conducted. Chinese scientists in the United States who have engaged in espionage or illegal technology transfer must be immediately deported. Action must be taken now because the success of the People's Liberation Army's first-generation biotechnology weapon, COVID-19, has only accelerated China's military ambitions. Next on John B. Wells' program, an interview with acclaimed author and columnist Diana West.